Okay, welcome to part 2. Guys, welcome to part 2. Now, in this second part here, we are looking at unguided Amali for the effect of light intensity on hydrilla. Guys, it's not easy to find the questions that I'm discussing. I mean, the previous one I discussed and as well as this. Okay, straight to the point. For this question whereby you have to provide procedure, okay, they mention your procedure. You can provide procedure as well, not a problem, but my more recommended answer would be you tackle it individually, MV, RV as well as constant, then precaution. On another video, I'll be discussing how to answer procedure. Okay, let's start. So, method to handle. Of course, before we start to go through, it's best you read through one round of what we are doing. Read through the experiment first. So, method to handle. Okay, the first one, which is the manipulated variable. Okay, as mentioned in the previous video, guys, you have to say, okay, let's use this. Use different, okay, I'm taking this, use different distance of light source, that is, sir, I do not know how much. Don't worry, all the values are here on the right. As you can see, 30, 40, 50, and 60. That is 20 cm, 30 cm, 40 cm, 50 cm, and 60 cm. Done. Moving on to, we have to use the table as well. Method to handle the responding variable. Guys, normally, similar to the previous video, normally we say measure and record, but here we cannot measure the air bubble. We have to count. Oops. Guys, count. Count and record the number of air bubbles. Okay, better follow this. Release in 5 minutes using a stopwatch. Guys, because 5 minutes we require to use this apparatus by using a stopwatch. And the last one, before the precautionary step, we just have to give one because it's a 4-mark question. Guys, for how to manage, how to handle the constant variable, fix the concentration of sodium hydrogen carbonate solution at 0.2, correct? Of 0.2 sodium hydrogen carbonate at 0.2%. At 0.2%, yeah. And last one, as for the precautionary step, ensure that hydrilla is fully immersed in the sodium hydrogen carbonate solution throughout the experiment. Okay, so guys, this is how you answer this part here. Moving on to, now because there are no values, I mean of course I can give you numbers, but to make it a little bit more challenging for you, I'll use alphabets instead. Guys, I'll use alphabets instead. So I'll just write A, B, C, D, E. If possible, please pause and think about what observation you would give. But I have to teach first. So, when the distance of light source is, okay, I take this, 20 cm. Guys, the number of air bubbles released in 5 minutes is A. Full stop. Full stop. So can I use 60 and then E? Can, no problem. But don't forget, in the first video I told you, after stating the values, probably it would be good to state higher or lower. So here, because 20 cm is closer to the hydrilla, so more air bubbles are released, which is higher. Okay, which is more or which is higher also can. Next, for C2, give an inference. Guys, inference, same thing, state the comparison and give a reason. So, more air bubbles are released because light intensity is higher. If you really want yourself to be super duper safe, leading to higher photosynthesis rate. Guys, this way your answer is really safe. Okay, I find it's a bit redundant for them to ask you MV, RV and also CV here, considering that they already asked you the method to handle in front, you know, it's like repetition, but never mind, guys. So we can just write distance of light source here. Next, we are looking at number of air bubbles released. You need not write 5 minutes, but if you really want to also, it's no problem. Release in 5 minutes. And the next one, you can say concentration of sodium hydrogen carbonate. I'm not too sure whether they will mark you wrong. Guys, I'm not too sure whether they will mark you wrong. I mean, just suddenly came to my mind. Because so far I haven't encountered question whereby they ask method to handle and after that they ask variable. So I'm guessing maybe this type, let's say for example, this type of question, maybe they want you to tally according to what you have written earlier. Means I'm not sure, guys. Maybe you write concentration here, but you write type of plant. Maybe wrong. Okay, maybe. So I think with that in mind, just in case if really this comes out, just tally accordingly. Okay, graph. Guys, we're not doing the graph, but just so you know, as the temperature. Okay, let's use another color. Oh, I think something orange. Oh, but orange I did just now. Green again. Okay, the graph that you should get would be number of air bubbles and distance. So as the distance increase, number of air bubbles reduce. You should get something like this. And guys, this is what I call a relationship question. Although they say state and inference, but actually this is relationship. They are trying to catch you whereby they want to make sure that you use back the MV and RV they give you. Here they didn't use distance, they never use bubble. They straight away use light intensity and rate of photosynthesis, which is the whole concept of this experiment. So guys, this is I think students' favorite like hypothesis. the higher the light intensity, the higher the rate of photosynthesis. And they did not mention explain. Guys, they did not mention explain, they just mention state. Nyatakan, tak perlu huraikan. Guys, tak perlu huraikan. Moving on, prediction question. Prediction question. So the experiment is repeated at 45 degrees Celsius. Predict the number of air bubbles released. My recommendation is that you use something from the table. So you have to decide whether you want to use A or E, because they never really mention the distance. So if temperature is higher, will it be lower than A for sure, which whereby the distance is the closest, most number of air bubble produced, or E? Definitely lesser than A. Okay, definitely lesser than A. Number of air bubbles. Number of air bubbles will be lesser than A. Why? When temperature is higher, enzymes start to denature. I just told a student earlier that human enzyme and plant enzyme is different. Our body temperature, the optimum is 37, right? Theirs is 20 something only, guys, 25, 26. Starts to denature. So rate of photosynthesis will reduce, and with that less oxygen gas is released. Okay, guys, that's all for this part. I think there will be a follow-up session after this to talk about how to write the procedure. Okay, guys, cheers. I'll catch you in the next video. Thank you. Bye bye.